Hi, I'm Kevin. I'm here to talk about how to get hassle-free AAV titer and empty full characterization on Stunner. Getting almost anything done in AAVs takes too much sample and time, until you try Stunner. Stunner delivers easy and efficient answers for AAV with empty full ratio and total capsid titer using only 2 microliters of sample. Let's dig in to learn more. You know the feeling where you just want a late night snack, so you start to dream about what to eat, but when you check the fridge, there's less there than you hoped? Sounds just like AAV characterization. That is exactly where Stunner can help you really know your AAV sample. When characterizing AAV, you have a lot of things going on. It's important to know how much protein is present, how much DNA is present, if things are intact, if they're the right size, and if there's any aggregation. It's a truly complex biologic that demands a lot from characterization techniques. That's why characterization of AAV is a very broad subject and is still evolving. For every one of these needs, there are many technologies trying to meet the need. Today I'll show you how Stunner can help address the needs shown in green. That's titers for full capsids and total capsids, empty full ratio, and if there's aggregation going on in an AAV sample. Let's focus in on two technologies, DLS and UV-Vis. Each of them tells a part of the story. DLS gets you info on capsid titer and aggregation, but can't say anything about what's happening with DNA. On the other hand, UV-Vis gets you the total amounts of protein in DNA and can link those to empty full ratio, but doesn't give you info about if any of this is contained in capsids. Each gets you valuable info, but neither tells you everything. So what if you could combine them to get a whole story? That's where Stunner brings it all together to deliver answers on capsid titer, empty full ratio, and aggregation with only 2 microliters of sample in less than a minute. And there's no labels, reagents, or standards. You just use the sample you've got. So how does Stunner do this? Simply add in 2 microliters of AAV above a titer of 10 to the 12th into the input well, and it's pulled into the microfluidic reservoir. At this step, the sample can sit for up to 2 hours before reading with no concerns for evaporation. After the plate is loaded into the instrument, the circuit is read first when empty, and that removes any absorbance contribution from the plastic, and then a pump applies a small vacuum which moves the sample into the microcuvettes to be read. Because the circuit has fixed path lengths, it can make very precise and accurate reads. Here we can see exactly what sample loading looks like with the help of some fluorescein and a blacklight. In 2 wells, we've already added 2 microliters of fluorescein. As you can see, to load another sample, you simply add in 2 microliters into that input well, and the sample wicks into the plate. Stunner contains 2 technologies to read each sample, UV-Vis for concentration and DLS for sizing. SLS intensity data is also read during a DLS experiment, and samples can be read from 1 to 96 at a time. Stunner's AAV quant application will combine all the pieces of information into the full story about your AAV. Checking out your AAV with DLS tells you what shape your sample is in, if there's only capsids, or if aggregates have started to ruin things. DLS also lets you figure out how much SLS intensity is coming from just your AAV capsids. That's just the green peak shown on the left graph, while the gray peak is aggregates and larger stuff. UV-Vis brings information on how much total protein is in your sample and how much single-stranded DNA is there as well. Based on some simple info you tell Stunner before an experiment, it calculates empty full ratio and percent full. What Stunner is doing is shown step by step here. First, DLS identifies the capsids, the aggregates, and how intense the signal is from each of those populations. In the middle, that info then helps you split up your SLS signal from a total into the amount just attributable to empty and full capsids combined. And finally, UV-Vis brings in that info about the empty full ratio plus protein and DNA totals to provide the last piece of the puzzle. Here's the whole story told in one graph. Starting with the left side, blue bar, which is the protein in the sample. The UV-Vis got you a total amount of protein, and here we're expressing it as how many possible capsids all that protein could make. By adding in DLS and SLS info, Stunner splits up that information into the light blue bar, which is aggregates and extra protein junk, and the dark blue bar, which is the total titer for the AAV capsids. On the right-hand side, in the green bar, the process follows the same steps for DNA. The total height of the green bar is the total amount of DNA expressed as the total number of viral genomes that you could make. Light green is extra DNA, and dark green is the number of full AAV capsids. Dividing dark green, which is full capsids, and dark blue, which is total capsids, gets you to a percent full number that we can also convert into empty full ratio for you. Here's that same data showing how Stunner helps you know your AAV inside out. Dark green divided by dark blue is percent full, which is shown in yellow, and purple is percent empty, which is just the remainder. Empty full ratio is percent empty divided by percent full, or purple divided by yellow. And if you prefer the term CP over VG, that will just be the inverse of the percent full number. So in collaboration with Stride Bio, we've compared Stunner versus ddPCR results for genome titer for two AAV types, single-stranded AAV on the left and self-complementary AAV on the right. The genome titer of the single-stranded AAV sample was characterized using a robust variety of ddPCR experiments to best capture the titer, while the tight error bars on Stunner come from a single run of triplicate samples. For the self-complementary AAV sample on the right, the optimized ddPCR protocol was used and compares very favorably to quantification by Stunner. For getting capsid titer, Stunner is way faster and requires less upfront work than ELISA's. Here we've taken full or empty AAV9 and ran a dilution series where we've compared Stunner's total capsid titer metric against the result for our capsid ELISA. The results have slopes close to 1 and R-squared values above 0.99. And importantly, it would take hours of hands-on work to get this ELISA data, while Stunner's done it in less than an hour with no sample prep and only about 5 or 10 minutes of actual hands-on work. Let's check out an example using Stunner. Here we have a sample of AAV5 from lot 1. In this case, DLS alone was already setting off alarm bells. There are a lot of aggregates shown by grade peaks in the DLS curve in the upper right. But let's look at Stunner's other readouts for this sample. Stunner has picked up on that messy DLS signal and used it to divide up the protein in DNA bars to show that there's a lot of protein and DNA in this sample that's not part of a capsid. Those are the light blue and light green bars. UV-Vis alone would have only told you the totals. You also needed DLS info to get the full story and see what's wrong. To keep going with this example, here we ran a second lot of that same AAV5 on Stunner. This time, the DLS looks pretty good, and the green capsid peak is most of the signal. When Stunner combines all the data together, it shows you that very little free or aggregated protein or DNA exists. Those dark blue and green bars go almost all the way to the top. So for this sample, we've confirmed aggregation is no problem, gotten total and full titers that show very little extra stuff in there, and have an empty full ratio too. That's a pretty good amount of data for just 1 minute and 2 microliters. To look more into our ability to see percent full values, we took stocks of empty and full AAV9 and mixed them at different ratios. For each combination, 100% full stock, 80% full stock, etc., Stunner gets a good read on the data, and percent full and percent empty align well with the expected values based on the dilutions that we used. This idea also works for any serotype. Just tell Stunner the basics about what serotype you have, amino acid sequence, or just what natural serotype you're working with, and what its DNA payload is, and Stunner takes care of the rest of the heavy calculating. Working with Stride Bio again, we examined how Stunner's quantification of percent full compares with AUC for a highly purified sample. AUC was read for the highest enriched percent full sample and dilutions with empty capsids used to create target percent full. AUC and Stunner correlate very well, and both are highly linear as shown by the high R-squared value. Importantly, AUC requires orders of magnitude more sample than Stunner and takes way longer than the 45 seconds a single Stunner read takes. If you try to fake out Stunner, here's what happens. In this case, we tried out 2 samples, one with a mixture of IgG and DNA to simulate full capsids, and another with just IgG to simulate empty capsids. Stunner knew what was there and wasn't fooled. On the left, Stunner says almost all the IgG in DNA is free and aggregated protein or free and aggregated DNA, which are the light blue and light green bars. And the story is similar on the right, where the light blue bars are almost the entire height of the graph. If Iodixanol is part of your process, it's useful to know exactly how much is in your sample and to check on the Iodixanol concentration after each buffer exchange to know how much buffer exchange you've completed. Stunner can help out here as well, and will detect Iodixanol in your sample as low as 0.0005% by volume. So from only 2 microliters, Stunner combines UV-Vis, DLS, and SLS data to tell the whole story about your AAV, delivering mountains of data about sample aggregation, AAV full and empty capsid titer, and empty-full ratio. So that's how Stunner delivers label-free, standard-free, and hassle-free gene therapy quant. So once you start using Stunner, your fridge that is supposed to be full of AAV characterization techniques will now contain at least one technique that you're really excited about. If you want to learn more about anything you saw here today, please reach out to us at info@unchainedlabs.com.